says the organogenesis story, but the organogenesis story began at MIT in the 1980s and had roller coaster rides up and down, and I think it would take too long and we'd starve and miss lunch. So really what I'm going to be talking about, as Michael said, is just some of the things that we've gone through in the last, I guess, 12 to 18 months as we've gotten our second cell therapy called Gentuit approved as a combination biologic device through the Office of Cell, Tissue, and Gene Therapy in CBER. So just a brief summary on organogenesis is that almost everything we do is comprised, it fits in this slide right now. We have two key products that we're focused on. Both are FDA approved. APLI as a Class 3 medical device through CDRH, and it's approved for venous leg ulcers and diabetic foot ulcers. And the second product, Gentuit, approved as a combination product through CBER. Similar technology platform, both allogeneic cells, keratinocytes, fibroblasts, seeded in a bovine collagen matrix. So when I'm asked to give talks, it's almost never on the regulatory side. So first of all, for the regulatory experts that are in the audience, I don't mean to be condescending because the talk might be a little bit superficial, and I don't suggest you know all of this, but I think the value of the talk is that sometimes when you develop product, it takes so many years before you get to that brief moment in time when you're before the FDA that it can seem a little bit like a black box. And for us, it seemed like a black box, and now we can look at it retrospectively, and we just want to share some of the learnings of the pre-license inspection and of the FDA audit and panel and so forth. Usually what I'm talking about is either our science, our clinical research, often manufacturing scale-up or commercialization, or I'm invited to sit on a panel like you just heard. But I think it's particularly relevant to talk about our regulatory experience right now. So that's what the focus will be. And the product called Gentuit is, as I said, keratinocytes, fibroblasts, and a collagen matrix. It's amazing for us who have just spent 20 years in chronic wounds that there are many people that can look at a gaping open wound and without distaste, but some people really get queasy when they look at the mouth. But to us now we're very acclimated. What you see here is a Gentuit application, and it looks complicated, but really to a periodontist or an oral maxillofacial surgeon, it's actually very, very easy, because what the standard of care is today is a palate graft, where you excise a piece of the patient's palate and you suture it on just like this, just like what we see here. And so the types of applications that Gentuit is appropriate for is in a non-submerged setting, anything from the replacement of a palate graft for a free gingival graft. It can be placed around teeth, around dental implants, around indentulous sites, things like oral cancer resection, and on and on. So in other words, when you need to induce site-specific tissue regeneration in a non-submerged application for adults. This will be the only clinical slide that I'll share with you, but it's the foundation for the approval, so it's the summary of the phase 3 clinical trial. And what you see is that the primary endpoint that FDA mandated was KT, which stands for keratinized tissue. Have you generated enough keratinized tissue? So in other words, have you solved the problem? And that's compared to the patient's own control. And interestingly, in the clinical trial, which is rare, is you can have a within-patient control. So half the mouth, on one side of the mouth, they had the standard of care, which is a palate graft, and on the other side, Gentuit, the cell therapy. And you're able to compare the primary endpoint, which is have you generated enough tissue, but also the secondary endpoints, which are relevant in order to build a product that is commercially successful, such as how is the color versus the adjoining tissue, what's the texture of it versus the other tissue, patient preference, is it sensitive, and of course pain. So what you can see is that the p-values were very, very strong for the primary endpoint. We generated far excess of the minimum that FDA mandated in terms of inducing soft tissue to regenerate. And the secondary endpoints of color, texture, patient preference were met. You'll also notice that what was not met was pain. And that actually, in a way, surprised us, but it shouldn't have, because the pain is generated by the palate graft. It's phase 3 trial is that the patients had 3 surgical procedures in the mouth simultaneously, and they weren't able to dissociate pain. They knew they felt pain, but they weren't able to dissociate it. So that will fall to subsequent phase 4 work. But very, very compelling clinical results. So that brought us to the FDA. And in general, these are the key milestones that we faced. And I think that the most important thing most people know here is that there is a PDUFA date where FDA is very much incentivized to get this whole process done in a 10-month timeline. Now, I think PDUFA 5, which is, if it's not out, is out shortly, is going to maintain a 10-month timeline, but only begin that when they've accepted the filing at day 60. So that's semantics, really. It's going to a 12-month filing. But some of these key milestones, the first one after day 60 of submission is the filing determination. And that can be a very bad day for a regulatory team if the FDA at day 60, having reviewed the file, deems that they're not going to go forward and look at it. The inspections, you can see, happen midway through the process. The primary review should generally be complete by month 8, and then 3 weeks prior to the end of the PDUFA date, the back and forth, that frequent and sometimes frantic negotiation between the company and FDA happens to do with post-marketing approval commitments and labeling and so forth. And it also has to be done 3 weeks ahead so that the final can work its way up CBER for ultimate approval at the highest levels. So these are the general timelines that we've had to look at. And I think that it's the point that I really want to make is it can take a decade, it can take more than a decade in our field, and it really gets concentrated down to a 10-month timeline. And even if you want to go deeper than that, when you get to panel, it's one day. So the Super Bowl happens every single year, but this might happen once in your career, once in the lifespan of the product. So it's an incredibly exciting but very stressful time to have basically packaged years and years and years of blood, sweat, and tears of data, and to be able to put it before the regulators in a very clear, concise, and compelling manner. And to do that, it's not only a huge resource in terms of effort, but also money. And I just think some of the costs, which not everybody realizes, is the $1.5 million BLA fee. Now, that doesn't apply to small companies of less than 500 employees, but nevertheless it does for those of us that are above 500 employees. And the consulting fees, I think not everybody appreciates. And that's really driven by the fact that no company does this every day. So there are consultants for which this is a well-traveled path, and really you need to share you really need to capture the learnings and to have them shepherd you through this. And that's whether it's a panel consultant, regulatory consultant, medical scientific consultants. But it's a very onerous and very expensive process. And then I think that the other point that I just highlight is that the FDA review team is very cross-functional, and these are the types of specialties that we face. And it's very important because, one way to say it is that, we live in the regenerative medicine world, and some of these specialties, they focus, the questions that they ask are centered around the expertise that they possess, and that they don't necessarily speak regenerative medicine, they speak, biopharmaceuticals or clinical pharmacology. Maybe a healthier way to say it is that, not that they don't speak regenerative medicine, but we don't speak their language, because ultimately we have to package everything we do and deliver it in a way that is compelling and understood by them, because their benchmarks typically are the types of language, of studies, of methodology, of design of the other biologic tox studies that they've looked at, not necessarily other cell therapies. But importantly, it really, it's a very professional relationship with the FDA, but it's not necessarily adversarial, because the FDA, once they've accepted it for review, really wants to be successful too. And those PDUFA dates really, really matter to them, is that they have performance goals to review and act on 90% of original BLAs in 10 months, and that they're incentivized based on meeting and achieving that. So I think that it's really in your interest and their interest if it can be a very collaborative effort, and to write the documents in proper format, clear, concise, and to try to understand what it is that they need, when they need it, how they need it. And there's a lot of things that we didn't necessarily see eye to eye on, but I guess, and one thing I could say is that the reviewers from CBER were very open-minded in hearing our argumentation, and often through the process we felt that we were having sway and influencing them. But I think when the dust settles, probably the biggest point is that the guidelines that they've issued for a reason, and they do tend, as much as they can be open and listen to our debate, they really did default largely to following the spirit, the philosophy of their guideline. So I mentioned out of that whole value chain that this is the area that I'll be focused on, but even these topics themselves could take all day. So I'm just going to talk about two of the most important exercises that were part of getting this CBER-approved product through. First of all, the pre-license inspection. So in terms of the pre-license inspection, there were 4 auditors that came to visit us, 2 from the Office of Cell, Tissue, and Gene Therapy, and then 2 with sort of a systems and GMP type background. And they stayed 9 to 5 for 5 days and met with all of our team. They reviewed 200 documents, interviewed 30 people, and it's just an incredible amount of effort that's required to put in. In the end, we mentioned 483s here. We had 9 observations. None of them were critical, and all were remediated prior to the subsequent approval. And that we deemed, actually, we were very satisfied with that. So in terms of the pre-license inspection, just the other thing is most of us, we do have commercialized products, we do have a day job, and the amount of effort from all of these departments was something that we underestimated. It touched everybody. I think that there were 60 people working for 1 year, so there was a really broad and deep effort to really capture all of the work and be able to get through this inspection. I should also note that APLI was approved as a Class 3 medical device. The clinical endpoints for Phase 3 trial really are no different, but on the preclinical, on the CMC side, on the manufacturing side, there are significant differences. So when we did a gap analysis, there's just a huge amount of gaps, and I think there were hundreds of gaps, and so that year of 60 people focusing on it was really a gap closing initiative where we were just going one by one to try to make sure that we adhered not only to CDRH standards but also to CBER standards. And it was a good catalyst for us because we entered this process as a Class 3 medical device company, and we exited it as both a biologics company as well as a medical device company. So we feel that we're stronger and that we adhere to the highest of all standards. We needed a project management team to guide us through all of these efforts, and the idea was to identify the management that were best capable to address the questions. I mentioned that 30 people were interviewed, so more than that had to be prepared. There had to be a bench. There had to be a whole team of people that were in a position to address it. And we did what was called mock audits. And the idea of mock audits is, I think it's kind of obvious that you need to do it, but you need to do it very, very, very rigorously, because I think that if you don't bring in the right people to go through and to look at you from the outside, you're going to miss some of those gaps. And really, the thing that we did really well was to identify the gaps, and then it's just an exercise of applying the proper resource to close them. So when actually the inspection came, and I mentioned it was 5 days, 9 to 5, there were a team of Organogenesis people in the room with the FDA, and this was a war room on the other side of the building that they were in. So there were FDA people asking questions. There were people from our quality and regulatory team addressing them, and there were people transcribing the questions, and the questions from the FDA would appear on the screen here in the war room, and people would run around and race to get in and get the questions. And when someone like me would walk in, you just get hip checked out of the way because people were just so focused on getting the FDA what they needed in the most timely, efficient manner. And it really does have a big impact to address the questions quickly, efficiently. So the second issue is the advisory committee. And having had a successful pre-license inspection, we progressed to the advisory committee. And if we thought the pre-license inspection was intimidating, the advisory committee really put you over the top. When I walked in the room and saw this that day, I was just so happy that Pat Bilbo, who's in the audience, was the quarterback of it, and not me, because it's designed to intimidate. But and when you think of who attends, it's a public meeting. You have the FDA there, you have their advisories, you have your competition, you have the media there. So it's an amazing, amazing show. And this is how it's set up, where you can see that the FDA and the advisory committee sit around that C-shaped panel with everybody else off in the audience, such as myself. And Pat and his team were sitting up at the top there, and we had some key opinion leader clinicians and scientific advisors that were in those white clear seats that could be called upon to address questions. So, and this is the type of timing that was required to make it work. I think that one of the key elements is the briefing package. And the briefing package, you can see how long it took us to put it together, but it's a concise 100 pages, and it's meant to represent everything that's relevant within the very broad BLA. So the amount of work, it's easy to provide information. It's very tough to make it concise, clear, compelling, credible. And when you think that your fate rests on not only the briefing document, but even just the executive summary of the briefing document, the amount of effort that has to be put in is amazing. And you can imagine that the remit of these advisors are to understand the BLA, to read the briefing document clearly, but let's just say that you better put a lot of effort into the executive summary, because it's really going to be the guide which orients them. So we had 3 mocks. We hired an outside consultant to guide us through this process, a company that just does this for a living, company after company after company, and can really guide on what the panel is supposed to function like, the types of questions that you could be prepared for. The first mock, what we did is we actually flew down from Boston to Houston for each of these mocks, and it's because we needed some of the key opinion leaders, the clinicians that ran the trials, and they couldn't displace themselves for all of these mocks because it was 3 different mock panels, each lasting 3 days, where there was preparation for the first days, and then the simulated mock was on day 3. And so we actually had to go to them, so we flew down there. But the goal was to bring tough, often ex-FDA panelists or the top people in the field and the various different scientific specialties, and just gruel us. And I guess it really demonstrated to us, we generated this data, we know this product, we've spent decades working on it, but it became very clear after the first mock that it would be terrible not to go through this process, because it's one thing to know the data, but to be able to package it and address it in a compelling way and handle the objections and speak the language of FDA is very, very onerous. So, I think we called the first mock shell shock, and then the second one we got a little bit stronger, and then a little bit stronger, with the mock 3 being really almost like a full-on dress rehearsal where we felt a lot more ready. So this is what happens, this is not exactly like every single one, but this is directionally what the panel looks like, is that there's a very brief introduction. We have to highlight, typically from an external key opinion leader, what the medical need is. We present the product efficacy, the safety, the risk-benefit, and then the conclusion. And you can see that life of this product is now, distilled down to minutes. And some of the main things is that because it's such a stressful environment, just to try to maintain focus, to warm up, to prevent distractions, and to really use the simplest persuasive language, to be credible but to be passionate about your case, and to answer clearly, succinctly, with conviction. And you don't have to just race to an answer. You can go and you can consult. It's okay to confer, but really also to correct any misinformation if and when it happens. Because one of the key things that was implored upon us is that the most important thing is to build trust capital. Don't try to be a salesman. Be passionate and with conviction, but don't be a salesman about it, because you're really out there trying to build that trust capital as well as to win every vote. So the type of thing just to give, I won't go into great detail, but just to highlight that in the first mock panel, we felt that we were a bit sloppy in how we addressed an issue such as the immunology of this allogeneic cell therapy. And so it was flagged, it was challenged in the mock. We researched, we practiced, we brought in top immunologists. We brought in the head of immunology from Oxford to sit in the panel, and she stood up and gave a very compelling recount of our immunology. And in the end, it was well accepted by panel. So, if we had just walked in without that, it's the same data. We didn't generate new data, but I think we were able to distill it in a way that managed the objection, and we otherwise may not have. So the amazing thing also is that you go through all this work, years and years and years, and in the end, they vote. And they vote right in front of you, and it's put up on the screen. So it's like a game show, where it looks like Family Feud, where after all of this, they vote on efficacy, and then they vote on safety. And you can see that the result was 15-0 vote on efficacy. So every one of those chairs is an actual panelist voting. And so that was green. And then for safety, 14-1 for safety. And the panelists that voted against safety was really focused on having longer-term safety data, which I think is relevant for all of us, because by the time you get, unless your program has been sufficiently delayed, you just won't have that long-term safety data when you find yourself in front of panels. So that's just an interesting note, but it wasn't for any other grander reason. So we were thrilled with this, and just to put it into perspective, for these panelists, as well as for the CBER folks that were doing the pre-license inspection, this was the first allogeneic cell therapy that they looked at. So it was very, very relevant. But to give some stats, Provenge, which we're all aware of from Dendreon, was 17-0 for safety and 13-4 for efficacy. LaVive, which is Fibrocell, also autologous, was 6-8 safety and 11-3 efficacy, and a 2,700 patient registry as a post-approval commitment. We have as a post-approval commitment to do a pediatric trial, and our approval and our label is very, very broad, but for adults only, and that makes sense because that's the only that we didn't study the pediatric population. So that's the team that were there that participated in it, and I think they look weary but happy there. That's Pat in the audience. I think he just recently stopped drinking champagne after months and months and months. But the eventual approval came in March 9th, 2012. And for us as a company, we got our venous leg ulcer indication in 1998, diabetic in 2001. So you can imagine, especially for those that have been brought up in the device industry, where you have new bells and whistles and products, this doesn't happen very often. This is a very big an initiative for us, and it took us many, many years to get there. So it was a well-deserved celebration. And the product is now available. It's going through what we call a soft launch, where we're training the top 40 to 50 periodontists, oral maxillofacial surgeons in 2012, with the hopes of a broader launch, leveraging them as our top key opinion leaders and advocates next year. So just very simply, I think that the point I've belabored is that a lot of effort comes down to a very short period of time, and that it requires a very, very professional approach, an approach that I don't think you can really possess completely in-house. Big companies leverage outside resources to get through this. And the mobilization, the dishevelment is absolutely enormous, and we certainly underestimated it. Working effectively with the FDA is absolutely critical. And then I think what's exciting about where we sit within Organogenesis is this was the catalyst to bring us into the biologics world. And when I look at our quality team, our regulatory team, our R&D team, they feel like they look like veterans who have been to war now, whereas perhaps a little more naive prior to this. So it was